Alright, in this recording, what we want to start to talk about is an overview of controlling gene expression. So again, the idea that we have all these genes, and we understand what a gene is now, how do we control what genes are on or off, right? So prokaryotes and eukaryotes must alter their gene expression in response to their changing environment. So again, expressing a gene means I'm going to take that information in DNA and start to go down the process of turning it into a protein that can do something. And in response to a changing environment, that may mean I need to do something to get away from something, or to eliminate a poison, or to bring in food. So multicellular organisms, we're going to expand on that, what we call differential gene expression, because those different genes on and off at different times and different intensities is also going to be responsible for growth and development. So different cell types. So it is an incredibly complex, what we call orchestra of gene expression. And so what you can see here is an embryo of a developing Drosophila fruit fly. And we'll hear a lot about Drosophila because it's a classic model organism. But each color here represents a different gene being turned on. And you can see the various patterns that form as different genes are turned on at different times for this thing to ultimately develop into a fly. Now, we're going to talk about both prokaryotic and eukaryotic gene expression in this course, and so you're going to need to keep them straight, the differences between them. In general, I'll speak to prokaryotic systems first because they tend to be simpler, but realize that the fundamental idea of the dogma is the same. The idea that you transcribe and translate is the same for both of these. So unless otherwise stated, things will be similar for both eukaryote and prokaryote, but there are definitely differences. Now, in bacteria, we often see a very, very efficient system. As we've talked about before, natural selection favors bacteria that produce only the products needed by that cell, right? So it's much more efficient than having all these tools, putting energy into making a bunch of tools that you don't need. Now, we're going to talk about this idea of feedback inhibition and gene regulation, and the Operon model for efficiency in prokaryotic life. So what we see here is the biochemical pathway for tryptophan synthesis. And tryptophan is one of our amino acids. So what you can see is if we have the raw material, a precursor for tryptophan, it can be processed by enzyme number 1, which is then handed off to enzyme number 2, which the product then is handed off to enzyme number 3, which finally makes tryptophan. Well, as tryptophan builds in the system, it can actually interact with enzyme 1 and act as an inhibitor. So what we have here is what's called feedback inhibition. The end product is acting to inhibit the production of itself. And that's how we control regulation. If we have a set amount, of some threshold amount, we don't need any more than that. We don't want to waste energy making more than we need. Now that's going to the purple arrow to the left. Now if we follow tryptophan on the blue arrow to the right, what we can see is that tryptophan can actually act in regulating the gene expression as well. So instead of just going and interfering with the active machinery, what it can do is actually say, well, I don't want to even make the machinery to begin with. So I'm going to down-regulate or shut down the genes that are responsible for making enzyme 1, 2, and 3 on the pathway. Right? And so this idea is called an operon, that all of those enzymes, 1, 2, and 3 in this case, are all under control of one operator. So they either all turn on at the same time, or they all turn off at the same time. And again, this is what we see in prokaryotes. We don't see this in eukaryotes. So we cluster genes that are functionally related. So enzymes 1, 2, and 3, you would never need just number 2 in prokaryotic biochemistry. It's only there if you're making tryptophan along this pathway. So you either need 1, 2, and 3, or you don't need any of them at all. So if we put them under one switch, it's more efficient to say, okay, let's turn on all of these genes all at the same time, or not. And so what we see is a cluster of these genes under control of what's called an operator. So this is the operon structure. You have your promoter in front of not just one coding sequence, but in this case, 3 different ones. And between that promoter and the first coding sequence, you see this binding site that we call the operator. And in this case, it's going to be the site for a repressor protein to bind to it and prevent RNA polymerase from proceeding down the DNA to do transcription. Okay? And we'll look at this in greater detail when in our next recording. Now, eukaryotic gene expression is a little bit more a lot more complicated. We're going to control responses to the environment, but also that environment is going to be the other cells around us as we grow and develop. And so that's going to be important in cell specialization and differential gene expression. So this idea of differential gene expression, different genes are turned on in different cells under different circumstances. This is a phrase that I'll use again and again and again. And the reason it's important is because every cell in your body and every eukaryotic organism has the same DNA as every other cell in that organism's body. All right? So let me say that one more time. So every cell in your body has a copy, basically, of your complete genome, of the trillion cells, right? So we cannot turn them on all at the same time. Clearly, there is something different between a skin cell and a liver cell. What's different? The difference must lie in that there are genes that are turned on in a skin cell that are not turned on in a liver cell, and vice versa, that result in a different morphology, or in essence phenotype, in those cells. So that's different genes being turned on. It's differential gene expression. Now, this is super important to control because when it doesn't go right, it leads to all kinds of diseases. Namely, cancer is one of the big ones. Now, eukaryotic gene expression, and we'll go over this in great detail over a couple different recordings, basically can be controlled at many, many different levels. And this allows for a very high degree of resolution of gene control, okay, of gene expression, controlling that. I can really dial that up or down along a continuum as opposed to just saying, oh, you're on or you're off. And we're going to see control at the chromosome level, gene expression literally making transcripts. We're going to see control at processing those transcripts, at letting those transcripts exit the nucleus, how long those transcripts will remain in the cytoplasm, how fast they're translated, how long the translation product remains, how long it's around, how it's processed to become active. So we'll address each of these in turn.